Chapter 5. What are big brains for? Or how culture stole our guts. By selectively attending to certain types of cultural content, like food, sex, and tools, and to particular models based on cues related to prestige, success, and health, individuals can most efficiently equip themselves with the best available cultural know-how. This acquired repertoire can then be honed and augmented by an individual's experience in the world. Crucially, however, these individually adaptive pursuits have an unintended consequence, which we saw when the MBAs were allowed to copy each other. The whole group gradually zeroed in on the optimal investment allocation. As individuals go about their business of learning from others in their group, the overall body of cultural information contained and distributed across the minds in the group can improve and accumulate over generations. To see more precisely how cumulative cultural evolution works, imagine a small group of forest-dwelling primates. One study visually represents this group along the top row, labeled Generation 0, with individuals represented by circles. One member of this generation has, on her own, figured out how to use a stick to extract termites from a termite mound. This trait is labeled T. Figuring this out is plausible for our ancestors, since modern chimpanzees do it. In Generation 1, Row 2, two of the offspring from Generation 1 copy the elder termite fisher because they note her success and are generally interested in things related to food. However, while copying this termite harvesting technique, one member of Generation 1 mistakenly infers that the stick his model was using had been sharpened, though actually it just broke funny when the model grabbed it. When the learner made his stick, he used his teeth to sharpen it to match his model stick. Elsewhere, another member of Generation 1 realizes he can use hollow reeds to drink from the water that gathers deep in the troughs of large trees. He uses this technique to obtain water when he crosses the savanna between patches of forest. In Generation 2, both the possessors of T2 and T star were preferentially copied, so their practices spread a bit. One member of Generation 2 even managed to pick up both T2 and T star, so she became particularly successful and was copied by 3 members of Generation 3. Then one day, an inattentive member of Generation 3 plunged his fishing stick into an old abandoned termite mound, not realizing the termites had long departed. Fortuitously, he happened to spear a rodent who had moved in after the termites left. Suddenly, the termite fishing stick became a general-purpose hole spear, now labeled T star star, which allowed this lucky fellow to tap new food sources as he started plunging his spear into every hole he could find. His success as a hole spearer meant that several members of Generation 4 attended to and learned from him. Meanwhile, another member of Generation 3, while goofing off, just happened to notice a rabbit enter his hole after a rain. While looking at the rabbit's tracks in the mud and thinking how tasty that rabbit would be, it struck her that she could look for tracks like these and follow them to locate active rabbit holes. This rabbit tracking is labeled T3. This was interesting, but not immediately useful, since she had no way to get the rabbit out of the hole. Nevertheless, years later, this mom showed the tracks to her daughter after they saw a rabbit. This happenstance was crucial, because the daughter had already learned T star star, hole spear. Now she could locate active rabbit holes and deploy her spear. A very useful technique. In Generation 5, while no one invented or lucked into anything, 3 members had learned T star star, T2, and T3. This package, a cultural adaptation, permitted them to spend more time on the savanna tracking rabbits to their holes, since they could also access water with T2, the straw. Soon, these primates began living at the edge of the forests, so they could hunt on the savanna. I'll call the combination of traits T star star, T2, and T3 the savanna hunting package. Keep in mind that this is a toy example meant to illustrate how selective cultural learning can generate a cumulative evolutionary process that generates cultural packages that are smarter than their bearers. My imaginary primates are better cultural learners than any living primate except us. Nevertheless, even if I had made them worse learners, I could have achieved the same endpoint with a larger population or more generations. Like us, or at least me, these primates were just bumbling myopically around their daily lives. Sometimes their mistakes led to innovations, and sometimes a rare circumstance thrust an insight upon someone who was otherwise goofing off. The key is that these occasional insights and lucky errors were preferentially passed on, persisted, and were eventually recombined with other traits to create a savanna hunting package. Now, consider this question. Is Generation 5 smarter than Generation 0? The former has better tools and can obtain food more efficiently. Later, we will see a variety of evidence suggesting that Generation 5 may in fact be smarter than Generation 0 if smartness is defined as an individual's ability to solve novel problems. Of course, there will be caveats and cautions. This imaginary ancestral primate crossed a crucial evolutionary threshold as it entered a regime of cumulative cultural evolution. This threshold is the point at which culturally transmitted information begins to accumulate over generations, such that tools and know-how get increasingly better fit to the local environments. This is the ratchet effect. It's the process that explains our cultural adaptations and, ultimately, the success of our species. As we'll discuss in Chapter 7, individuals reliant on cultural adaptations often have little or no understanding of how or why they work, or even that they are doing anything adaptive. The central argument in this book is that relatively early in our species' evolutionary history, perhaps around the origins of our genus, Homo, about 2 million years ago, we first crossed this evolutionary Rubicon, at which point cultural evolution became the primary driver of our species' genetic evolution. This interaction between cultural and genetic evolution generated a process that can be described as autocatalytic, meaning that it produces the fuel that propels it. Once cultural information began to accumulate and produce cultural adaptations, the main selection pressure on genes revolved around improving our psychological abilities to acquire, store, process, and organize the array of fitness-enhancing skills and practices that became increasingly available in the minds of the others in one's group. As genetic evolution improved our brains and abilities for learning from others, cultural evolution spontaneously generated more and better cultural adaptations, which kept the pressure on for brains that were better at acquiring and storing this cultural information. This process will continue until halted by an external constraint. I call the threshold between typical genetic evolution and the regime of autocatalytic culture-driven genetic evolution the Rubicon. During the Roman Republic, the muddy red waters of the river Rubicon marked the boundary between the province of Cisalpine Gaul and Italy proper, which was administered directly by Rome itself. Provincial governors could command Roman troops outside of Italy, but under no circumstances could they enter Italy at the head of an army. Any commander who did this, and any soldiers who followed him, were immediately considered outlaws. This rule had served the old republic well until, in 49 BC, Julius Caesar crossed the Rubicon at the head of the loyal Legio XIII Gemina, the 13th Twin Legion. For Caesar and this legion, there was no turning back after crossing the Rubicon. Civil war was inevitable, and Roman history would be forever altered. Similarly, in crossing our evolutionary Rubicon, the human lineage embarked down a novel evolutionary path from which there was no turning back. To see why there's no turning back, imagine being a member of Generation 6. Should you focus on inventing a new trait, or on making sure you accurately locate and copy those individuals with T**, T2, and T3? You might invent a good trait as adaptive as T, but you'll never invent something as good as the Savannah hunting package of T** plus T2 plus T3. Thus, if you don't focus on cultural learning, you will lose out to those who do. As the process continues over generations, the selection pressures only increase. The more culture accumulates, the greater the selection pressures on genes for making one an adept cultural learner with a bigger brain capable of harnessing the ever upward spiraling body of cultural information. Consider the memory, or brain storage space, required by our 6 different generations. In Generation 0, at most, you could invent 1 trait in your lifetime, so you only need brain space for 1 trait. However, by Generation 5, you'd better have storage space in your brain for T**, T2, and T3, and you'd best know how they fit together. The memory space demanded in Generation 5, if one wants any chance of out-surviving and out-reproducing others in the population, has increased 3-fold in only 6 generations. If genes spread that expand the brains of Generation 6, the selection pressure for bigger and better brains won't abate because cultural evolution will continue to expand the size of the cultural repertoire, of the body of know-how one could learn if one were sufficiently well equipped. This culture-gene co-evolutionary ratchet made us human. We've already seen some of the evidence that culture drove human evolution. In Chapter 2, we discussed that when toddlers competed against other apes in a variety of cognitive tasks, the only domain in which they kicked butt was social learning. Otherwise, for quantities, causality, and space, it was pretty much a tie. That's precisely what you'd expect if culture drove the expansion of our brains, honed our cognitive abilities, and modified our social motivations. In Chapter 3, by accompanying various hapless explorers, we saw that our species' ability to live as hunter-gatherers depends on acquiring the local cultural knowledge and skills. And in Chapter 4, we explored how natural selection has shaped our psychology to allow us to selectively target and extract adaptive information from our social milieu. Moving forward, the following list summarizes some of the products of culture-gene coevolution that I cover in this book. To begin, here I first examine 5 ways in which cultural evolution has influenced and interacted with genetic evolution to shape our bodies, brains, and psychology. To understand the information, I'll first read Culturally Transmitted Selection Pressure. These are features of the world that were created by cultural evolution, but that subsequently had consequences for genetic evolution. given in the next set of information, co-evolved genetic consequence, and sparked a coevolutionary duet between genes and culture. Examples of how cultural evolution and its products have shaped human genetic evolution. Culturally transmitted selection pressure. Cumulative culture. Accumulating body of cultural knowledge creates dependence. Co-evolved genetic consequences. Specialized cultural learning abilities for selectively acquiring adaptive information from others. Long childhoods and larger brains prepared for cultural learning and practice with extensive brain wiring over decades. Other implications: selection pressure for greater sociality, difficult childbirth due to oversized heads, demands for more child care. We touch on these topics in chapters 2 through 5, 7 and 8, and 16. Culturally transmitted selection pressure. Food processing, cooking, leaching, pounding, and chopping. Co-evolved genetic consequences. Increasing dependence on processed food, including cooked foods, results in small teeth, gapes, mouths, colons, and stomachs. Possible interest in fire during childhood. Other implications: frees energy for brain building and favors the sexual division of labor. We'll touch on these topics in chapters 5 through 7, 12, and 15. Culturally transmitted selection pressure. Persistence hunting, tracking, water containers, and animal behavioral know-how. Co-evolved genetic consequences. Distance running facilitated by springy arches, slow twitch muscle fibers, shock-enforced joints, a nuchal ligament, and innervated eccrine sweat glands. Other implications: human lineage becomes high-level predator. We'll discuss these topics in Chapter 5. Culturally transmitted selection pressure. Folk biology, increasing knowledge about plants and animals. Co-evolved genetic consequences. Folk biological cognition, hierarchical taxonomies with essentialized categories, category-based induction, and taxonomic inheritance. Other implications: universal tree-like taxonomies for categorizing the natural world. We'll talk about these topics in chapters 5 and 7. Culturally transmitted selection pressure. Artifacts, increasingly complex tools and weapons. Co-evolved genetic consequences. Anatomical changes to hands, shoulders, and elbows. Direct cortical connections into spinal cord. Artifact cognition, functional stance. Other implications: greater manual dexterity and throwing abilities, increased physical weakness. We'll touch on these topics in chapters 5, 12, 13, and 15. Culturally transmitted selection pressure. Wisdom of age. Opportunities to use and transmit culture gleaned over a lifetime. Co-evolved genetic consequences. Changes in human life history, extended childhood, adolescence, and a longer post-reproductive lifespan, menopause. Other implications: cooperation in child investment and rearing. We'll talk about these topics in chapters 4, 5, 8, 12, 15, and 16. Culturally transmitted selection pressure. Complex adaptations. Pressure for high-fidelity cultural learning. Co-evolved genetic consequences. Sophisticated abilities to infer others' mental states, theory of mind or mentalizing, and over-imitation. Other implications: dualism, a preparedness to understand minds without bodies. We'll touch on these topics in chapters 4, 7, 12, and 13. Culturally transmitted selection pressure. Information resources, variation in skill or know-how among individuals. co-evolved genetic consequences, prestige status, suite of motivations, emotions, and ethological patterns that produce a second type of status. Other implications, prestige-based leadership and greater cooperation. We talk about these topics in chapters 4 and 8. Culturally transmitted selection pressure, social norms, enforced by reputations and sanctions. Co-evolved genetic consequences, norm psychology, concerns with reputation, internalization of norms, pro-social biases, shame and anger at norm violators, cognitive abilities for detecting violations. Other implications, strengthens effect of intergroup competition on cultural evolution. We'll talk about these topics in chapters 9 through 11. Culturally transmitted selection pressure, ethnic groups, culturally marked membership across social groups. Co-evolved genetic consequences, folk sociology, in-group versus out-group psychology that cues off phenotypic markers, which influence cultural learning and interaction. Other implications, tribal and ethnic groups, later nationalism, and parochial religions. We'll talk about these in Chapter 11. Culturally transmitted selection pressure, languages, transmitted gestures and vocalizations. Co-evolved genetic consequences, changes in throat anatomy, audio processing, specialized brain regions, and tongue dexterity. Other implications, massive increase in the rate of cultural transmission. We discuss this in Chapter 13. Culturally transmitted selection pressure, teaching, opportunities to facilitate cultural transmission. Co-evolved genetic consequences, communicative or pedagogical adaptations, white sclera, whites of the eyes, eye contact, pedagogical inclinations, etc. Other implications, higher fidelity transmission and more rapid cultural evolution. We'll talk about this in Chapter 13. Now, let's examine how crossing the evolutionary Rubicon into a regime of cumulative cultural evolution helps explain several of our species' characteristics. Big brains, fast evolution, and slow development. Compared to those of other animals, our brains are big, dense, and groovy. While we don't have the biggest brains in the natural world, whales and elephants beat us, we do have the most cortical interconnections and the highest degree of cortical folding. Cortical folding produces that crumpled wad of paper, groovy appearance that particularly characterizes human brains. But that's just the beginning of our oddities. Our brains evolved from the size of a chimpanzee's at roughly 350 cubic centimeters to 1,350 cubic centimeters in about 5 million years. Most of that expansion, from about 500 cubic centimeters upward, took place only in about the last 2 million years. That's fast in genetic evolutionary terms. This expansion was finally halted about 200,000 years ago, probably by the challenges of giving birth to babies with increasingly bulbous heads. In most species, the birth canal is larger than the newborn's head, but not in humans. Infant skulls have to remain unfused in order to squeeze through the birth canal in a manner that isn't seen in other species. It seems our brains only ceased expanding because we hit the stops set by our primate body plan. If babies' heads got any bigger, they wouldn't be able to squeeze out of mom at birth. Along the way, natural selection came up with numerous tricks to circumvent this big-headed baby problem, including intense cortical folding, high-density interconnections, which permit our brains to hold more information without getting bigger, and a rapid post-birth expansion. Specifically, newborn human brains continue expanding at the faster pre-birth gestational rate for the first year, eventually tripling in size. By contrast, newborn primate brains grow more slowly after birth, eventually only doubling in size. After this initial growth spurt, our brains continue adding more connections for holding and processing information, new glial cells, axons, and synapses, over the next 3 decades of life, and even beyond, especially in the neocortex. Consider our white matter, and specifically the process of myelination. As vertebrate brains mature, their white matter increases as the axonal connections among neurons are gradually burned in and wrapped in a performance-enhancing coating of fat called myelin. This process of myelination makes brain regions more efficient, but less plastic, and thus less susceptible to learning. To see how human brains are different, we can compare our myelination with that of our closest relatives, chimpanzees. For the cerebral cortex, one study shows the fraction of myelination, as a percentage of the adult level, during 3 different developmental periods: 1, infancy; 2, childhood, called the juvenile period in primates; and 3, adolescence and young adulthood. Infant chimpanzees arrive in the world with 15% of their cortex already myelinized, whereas humans start with only 1.6% myelinized. For the neocortex, which has evolved more recently and is massive in humans, the percentages are 20% and 0%, respectively. During adolescence and young adulthood, humans still have only 65% of their eventual myelination complete, whereas chimpanzees are almost done at 96%. These data suggest that, unlike chimpanzees, we continue substantial wiring up into our 3rd decade of life. Human brain development is related to another unusual feature of our species, our extended childhoods, and the emergence of that memorable period called adolescence. Compared to other primates, our gestational and infancy periods, birth to weaning, have shortened, while our childhoods have extended, and a uniquely human period of adolescence has emerged prior to full maturity. Childhood is a period of intensive cultural learning, including playing and the practicing of adult roles and skills, during which time our brains reach nearly their adult size, while our bodies remain small. Adolescence begins at sexual maturity, after which a growth spurt ensues. During this time, we engage in apprenticeships as we hone the most complex of adult skills and areas of knowledge, as well as build relationships with peers and look for mates. The emergence of adolescence and young adulthood has likely been crucial over our evolutionary history, since in hunting and gathering populations, hunters do not produce enough calories to even feed themselves, let alone others, until around age 18, and won't reach their peak productivity until their late 30s. Interestingly, while hunters reach their peak strength and speed in their 20s, individual hunting success does not peak until around age 40, because success depends more on know-how and refined skills than on physical prowess. By contrast, chimpanzees, who also hunt and gather, can obtain enough calories to sustain themselves immediately after infancy ends, around age 5. Consistent with our long period of wiring up, this pattern and contrast with chimpanzees reveals the degree to which we humans are dependent on learning for our survival as foragers. Our unusually big brains, with their slow neurological and behavioral development but rapid evolutionary expansion, is precisely what you'd expect if cumulative cultural evolution had become the driving selection pressure in the evolution of our species. Once cumulative cultural evolution began to produce cultural adaptations, like cooking and spears, individuals whose genes have endowed them with the brains and developmental processes that permit them to most effectively acquire, store, and organize cultural information will be the most likely to survive, find mates, and leave progeny. As each generation gets brains that are a little bigger and a little better at cultural learning, the body of adaptive know-how will rapidly expand to fill any available brain space. This process will shape the development of our brains by keeping them maximally plastic and programmed to receive, and our bodies by keeping them small and calorically inexpensive until we've learned enough to survive. This culture-gene coevolutionary interaction creates an autocatalytic process such that no matter how big our brains get, there will always be much more cultural information in the world than any one of us can learn in a lifetime. The better our brains get at cultural learning, the faster adaptive cultural information accumulates, and the greater the pressure on brains to acquire and store this information. This view also explains three puzzling facts about human infants. First, compared to other species, babies are physically altricial, meaning that they are weak, under-muscled, fat, and uncoordinated. Sorry, babies, but it's true. By contrast, some mammals exit the womb ready to walk, and even primates rapidly figure out how to hang on to mom. Meanwhile, above the neck, human babies' brains are developmentally advanced at birth compared to those of other animals, having passed more of the mammalian neurological landmarks than other species. Fetuses are already acquiring aspects of language in the womb, reference chapter 13, and babies arrive ready to engage in cultural learning. Before they can walk, feed themselves, or safely defecate, infants are selectively learning from others based on cues of competence and reliability, reference chapter 4, and can read others' intentions in order to copy their goals. Third, despite being otherwise developmentally and cognitively advanced, babies' brains arrive highly plastic, unmyelinized, and continue to expand at their gestational rate. In short, while being otherwise nearly helpless, babies and toddlers are sophisticated cultural learning machines. Natural selection has made us a cultural species by altering our development in ways that, 1, slowed the growth of our bodies through a shortened infancy and extended childhood, but added a growth spurt in adolescence, and 2, altered neurological development in complex ways that make our brain advanced at birth, yet both highly expandable and enduringly plastic. As we go along, I will consider how our fast genetic evolution, big adult brains, slow bodily development, and gradual wiring up are made possible only as part of a larger package of features that include the sexual division of labor, intensive parental investment in children, and the long post-reproductive lives we associate with menopause. These features of our species will interact in crucial ways with cultural evolution. Food processing externalizes digestion. Compared to other primates, humans have an unusual digestive system. Starting at the top, our mouths, gapes, lips, and teeth are oddly small, and our lip muscles are weak. Our mouths are the size of a squirrel monkey's, a species that weighs less than 3 pounds. Chimpanzees can open their mouths twice as wide as we can, and hold substantial amounts of food compressed between their lips and large teeth. We also have puny jaw muscles that reach up only to just below our ears. Other primates' jaw muscles stretch to the tops of their heads, where they sometimes even latch onto a bony central ridge. Our stomachs are small, having only a third of the surface area that we'd expect for a primate of our size, and our colons are too short, being only 60% of their expected mass. Our bodies are also poor at detoxifying wild foods. Overall, our guts, stomachs, small intestines, and colons are much smaller than they ought to be for our overall body size. Compared to other primates, we lack a substantial amount of digestive power all the way down the line, from our mouths' inability to break down food to our colon's capacity to process fiber. Interestingly, our small intestines are about the size they should be, an exception that we'll account for in a moment. How can culture explain this strange physiological feature of humans? The answer is that our bodies, and in this case our digestive systems, have co-evolved with culturally transmitted know-how related to food processing. People in every society process food using techniques that have accumulated over generations, including cooking, drying, pounding, grinding, leaching, chopping, marinating, smoking, and scraping. Of these, the oldest are probably chopping, scraping, and pounding with stone tools. Chopping, scraping, and pounding meat can go a long way because they tenderize by slicing, dicing, and crushing the muscle fibers, partially replacing some of the functions of teeth, mouths, and jaws. Similarly, marinades initiate the chemical breakdown of foods. Acidic marinades, such as that used for the coastal South American dish ceviche, begin literally breaking down meat proteins before they reach your mouth, mimicking the approach taken by your stomach acid. And as we discovered with Nardoo, leaching is one of a host of techniques that hunter-gatherers have long used to process food and remove toxins. Of all these techniques, cooking is probably the most important piece of cultural know-how that has shaped our digestive system. The primatologist Richard Wrangham has persuasively argued that cooking, and therefore fire, has played a crucial role in human evolution. Richard and his collaborators laid out how cooking, if done properly, does an immense amount of digestion for us. It softens and prepares both meat and plant foods for digestion. The right amount of heating tenderizes, detoxifies, and breaks down fibrous tubers and other plant foods. Heating also breaks down the proteins in meat, dramatically reducing the work for our stomach acid. Consequently, by contrast with meat-eating carnivores, for example, lions, we do not often retain meat in our stomachs for hours because it typically arrives partially digested by pounding, scraping, marinating, and cooking. While all this food processing reduces the digestive workload of our mouths, stomachs, and colons, it does not alter the need to actually absorb the nutrients, which is why our small intestines are about the right size for a primate of our stature. What is often underemphasized in this account is that food processing techniques are primarily products of cultural evolution. Cooking, for example, is not something we instinctually know how to do, or even can easily figure out. If you don't believe me, go outside and make a fire without using any modern technology. Rub two sticks together, make a fire drill, find naturally occurring flint or quartz, etc. Put that big brain to work. Maybe some fire instincts designed by natural selection to solve this recurrent dilemma of our ancestral environments will kick in and guide you. No luck? Unless you've had training, that is, received cultural transmission, it's very unlikely you were successful. Our bodies have been shaped by fire and cooking, but we have to learn from others how to make fire and cook. Making fire is so unnatural and technically difficult that some foraging populations have actually lost the ability to make fire. These include the Andaman Islanders, off the coast of Malaysia, Siriano, Amazonia, northern Aceh, and perhaps Tasmanians. Now, to be clear, these populations couldn't have survived without fire. They retained fire, but lost the ability to start new fires on demand. When one band's fire was inadvertently extinguished, say during a fierce storm, they had to head off to locate another band whose fire had not gone out, hopefully. However, living in small and widely scattered groups in frosty Paleolithic Europe, the fires of our bigger-brained Neanderthal cousins probably sometimes went out and weren't to be reignited for thousands of years. In Chapter 12, we'll discuss how and why such important losses are not surprising. It's likely that our species' reliance on fire began with the control of fire, perhaps obtained from naturally occurring sources. Nevertheless, just capturing, sustaining, and controlling fire requires some know-how. Keeping a fire going may sound easy, but you have to keep it going all the time, during rainstorms and high winds and while traveling over long distances. I learned something about this while living in the Peruvian Amazon among an indigenous group called the Matsugenka. After transporting what looked like a dead, charred log to her distant garden, I saw a Matsugenka woman breathe life back into a hidden ember using a combination of dried moss, which she brought with her, and thermal reflection from other logs. I was also embarrassed when another young Matsugenka woman, with the requisite infant slung at her side, stopped by my house in the village to rearrange my cooking fire. Her adjustments increased the heat, created a convenient spot for my pot, reduced the smoke and my choking, and eliminated much of the need for my constant tending. Cooking is also difficult to learn through individual trial and error experience. For cooking to provide a digestive aid, it has to be done right. Bad cooking can actually make food harder to digest and increase its toxins. And what constitutes effective cooking depends on the type of food. With meat, doing the most obvious thing, to me, of placing pieces right in the flames can lead to a hard, charred outside and a raw interior, exactly what you don't want. Consequently, small-scale societies have complex repertoires of food processing techniques that are specific to the food in question. For example, the best cooking technique for some foods involves wrapping them in leaves and burying them in the fire's ash for a long time. How long? Meanwhile, many hunters eat the liver of their kill raw, on the spot. Livers, it turns out, are energy-rich, soft, and delicious when eaten raw, except for those species in which eating the liver can be deadly. Do you know which those are? Inuit hunters don't eat polar bear livers raw because they believe such livers are toxic. And they are correct, according to laboratory research on the question. The rest of the kill is typically butchered, sometimes pounded, possibly dried, and then cooked, though different parts of the kill are cooked in different ways. The impact of this culturally transmitted know-how about fire and cooking has had such an impact on our species' genetic evolution that we are now, essentially, addicted to cooked food. Wrangham reviewed the literature on the ability of humans to survive by eating only raw foods. His review includes historical cases in which people had to survive without cooking, as well as studies of modern fads, such as the raw foods movement. The long and short of all this is that it's very difficult to survive for months without cooking. Raw foodists are thin and often feel hungry. Their body fat drops so low that women often stop menstruating or menstruate only irregularly. This occurs despite the supermarket availability of a vast range of raw foods, the use of powerful processing technologies like blenders, and the consumption of some pre-processed foods. The upshot is that human foraging populations could never survive without cooking. Meanwhile, apes do just fine without cooking, though they do love cooked foods. Our species' increasing dependence on fire and cooking over our evolutionary history may have also shaped our cultural learning psychology in ways that facilitated the acquisition of know-how about fire making. This is a kind of content bias in our cultural learning. The UCLA anthropologist Dan Fessler argues that during middle childhood, ages 6 to 9, humans go through a phase in which we are strongly attracted to learning about fire by both observing others and manipulating it ourselves. In small-scale societies, where children are free to engage this curiosity, adolescents have both mastered fire and lost any further attraction to it. Interestingly, Fessler also argues that modern societies are unusual because so many children never get to satisfy their curiosity, so their fascination with fire stretches into the teen years and early adulthood. The influence of socially learned food processing techniques on our genetic evolution probably occurred very gradually, perhaps beginning with the earliest stone tools. Such tools had likely begun to emerge by at least 3 million years ago, reference chapter 15, and were probably used for processing meat, pounding, chopping, slicing, and dicing. Drying meat or soaking plant foods may have emerged at any time, and probably repeatedly. By the emergence of the genus Homo, it's plausible that cooking began to be used sporadically, but with increasing frequency, especially where large fibrous tubers and meat were relatively abundant. Our repertoire of food processing methods altered the genetic selection pressures on our digestive system by gradually supplanting some of its functions with cultural substitutes. Techniques such as cooking actually increase the energy available from foods and make them easier to digest and detoxify. This effect allowed natural selection to save substantial amounts of energy by reducing our gut tissue, the second most expensive tissue in our bodies, next to brain tissue, and our susceptibility to various diseases associated with gut tissue. The energy savings from the externalization of digestive functions by cultural evolution became one component in a suite of adjustments that permitted our species to build and run bigger and bigger brains.